Hello everybody, this is the Biology Trial 2 exam review for the semester 2 for grade 9 at the International School of Innovation. So, chapter 1, activity 1, it's about our food. So, what is the food that we eat? What does it contain? What happens to the food when we eat in our body? What is the final fate of the food? To start with, we have to talk a little bit about the definition of digestion. Digestion, simple definition, is the transformation of complex food molecules into the simplest one. This is the complex food molecules. You are digested into simplest one used to be used by the body cells. Mainly these are called nutrients. So, which system is responsible for the digestion of food? As we know, it's the digestive system for sure. So, digestive system mainly is divided into two main branches. The first branch is called digestive tube, and the second is called digestive gland. What is the digestive tube? It's where the organs through which food can pass through, while digestive glands is organs through which food doesn't pass through. Organs of the digestive tube and the digestive glands. Digestive tube, we have the mouth, then we have the esophagus, stomach, small intestine, large intestine, and finally we have the anus. While digestive glands, we have salivary gland, gastric gland, the stomach, pancreas, liver, and we have intestinal cells or intestinal glands, sorry. So let's move here to the function of the digestive gland. Digestive glands, they are known to secrete digestive juices into their site of action. What is this? Digestive gland. First, salivary glands. What do they secrete? They secrete saliva, where at the level of the mouth, where it is neutral, meaning that pH is equal to 7. Gastric glands, they secrete gastric juice at the level of the stomach, where it's acidic, meaning that pH is less than 7. Pancreas secretes pancreatic juice at the level of the small intestine. Liver secretes bile at the level of the small intestine. Intestinal glands, they secrete intestinal juice also at the level of the small intestine. Those three, they are the level of the small intestine. The medium is basic, meaning that pH is greater than 7. So, let's move now to what the meaning of neutral. Neutral medium is meaning the mouth. Salivary gland, they secrete the saliva at mouth. Mouth is neutral, where pH is equal to 7. Acidic, pH is less than 7. Basic, pH is greater than 7. Foods, they are divided into organic and inorganic. Example about organic: carbohydrates, proteins, lipids, and vitamins, while inorganic, water and mineral salts. Let's move to this table. Category of organic food. We have mainly, while talking grade 9, about carbohydrates, about proteins, and about lipids. I'm going to talk about the complex form, the intermediate, and the simplest form in the coming slides. So now we can go deeply into the definition of digestion. So digestion is the set of chemical and mechanical transformation of complex food molecules into simpler nutrients. These nutrients, they are going to be used by the body cells to produce energy needed for the activities of the cell, which is known by the cellular activity. Category of organic food. Pay attention to this. You have to study this table. You have to know every category of organic food. Which one is the complex, which one is the intermediate, and which one is the simplest form of food. So carbohydrates, they are natural source of sugars, fat sources of energy. They contain carbon, hydrogen, and oxygen. The complex form of carbohydrates is known to be the polysaccharides. Polysaccharides is the complex chain of simple sugar. We have starch is an example. We have cellulose, which is example of carbohydrates in plants, and we have glycogen in animals. Intermediate is known to be di. Di meaning that too. So it's contained two simple sugars. They are bounded together. First example is maltose. It's glucose plus glucose. Second, we have lactose. It's glucose plus galactose, mainly in milk. Sucrose is glucose plus fructose in fruits. Last one, simplest form or the nutrient form, it's the monosaccharide. Mono, it's only one. So it's made up of one unit only. Example, glucose, fructose, galactose, all of them, they are reducing sugar. Moving to proteins. Proteins, they are called plastic food. Proteins, they are complex chain of more than 52 amino acids. Polypeptides, this is the intermediate, it's less than 52 amino acid, while the simplest form is the simplest form, which is made up of only one unit. Lipids, they are very energetic. The same quantity of lipids have a double amount of energy that's supplied by carbohydrates. Complex form is lipids. Intermediate, there is no intermediate, and the simplest form is fatty acid plus glycerol. How to identify the presence of certain categories of food. First, we have food test. What is the aim and step of the test? What is the positive result and what is the negative result? And finally, analysis of the positive and the indication. Let's start one with water test. Water test, from its name, it's aimed to identify the presence of water. Steps of the test, you have to heat the food in the test tube. We have to heat it. Positive result is the presence of water droplets on the wall of the test tube. This is a positive result. While if it's a negative, no water droplets are formed. So more water droplets at the walls of the test tube. Analysis. After heating the food, water droplets are formed on the walls of the test tube. What does this indicate? This indicates the presence of water in the food. Test number 2 is salt test. Salt test, it aims to identify the presence of sodium chloride. We have to add distilled water to the food in a test tube and we have to shake it well. Add silver nitrate AgNO3 to the test tube. White precipitate is going to appear, which is called silver chloride crystals. Negative result, no white precipitate will appear. What does this analysis? Analysis after adding distilled water and silver nitrate solution to the food, white precipitate is going to appear. What does this indicate? This indicates the presence of salt in the food. Number 3, pay attention to this one, Fehling test. Fehling test is mainly for the reducing sugar, glucose, fructose, galactose, maltose, lactose, and so on. So we have to add equal numbers of Fehling, NaOH, and Fehling B, CuSO4. Heat the contents of the test tube. Positive result, brick red precipitate appears. Negative, solution is going to remain blue in color. Analysis, after adding equal drops of Fehling A, which is NaOH, and Fehling B, CuSO4, and applying them to Fehling test, brick red precipitate is going to appear. What does this indicate? This indicates the presence of reducing sugar in the food. So brick red precipitate is an indication of the presence of reducing sugar. Iodine test, which is test number 4. Iodine test is aimed to the presence of starch. What do we have to do? We have to add equal drops or few drops, of iodine solution. If dark blue color appears, this is a positive result. If no dark blue color, mean that orange, yellow, brown, or yellowish color mainly, so it's a negative result. After adding iodine to the food, dark blue color appears. What does this indicate? This indicates the presence of starch in the food. Pyrite test. Pyrite test is done for the presence of proteins and or polypeptides. How can we do this test? We can do it by adding equal numbers of drops of NaOH, which is colorless, and CuSO4, which is blue in color. What is the positive result? Violet color is going to appear. Negative result, color is going to remain blue in color. What is the analysis? After adding equal drops of NaOH and CuSO4, or after applying pyroTest, violet color. So the key here is the violet. So proteins with pyroTest, they're going to give violet color. What does this indicate? This indicates the presence of protein and or polypeptides. And pay attention to this one, and or polypeptides. Moving to coagulation test. It aims to the presence of proteins. Heat food till boiling. If it's coagulate, so it's a positive result. If it doesn't, so it's a negative result. After heating till boiling, the food is going to coagulate. This indicates the presence of proteins. Translucent spot test. It aims to test the presence of lipids. What we have to do is to rub a piece of food against a paper. Observe it under the sunlight. If a translucent spot appears, this is a positive result indicating the presence of lipids. No translucent, so no lipids. After rubbing the food against the paper and observing it under sunlight, a translucent spot is formed. What does this indicate? This indicates the presence of lipids in food. Sodium test. Sodium test indicates the presence of lipids. We have to add sodium test solution. A pink color is going to appear. It's a positive result indicating the presence of lipids. If no pink color, so no lipids. After applying sodium test, pink color appears. What does this indicate? This indicates the presence of lipids in food. So these tests, you have to study them. You have to know the positive result and the negative result. What does each test aims for? It aims for the presence of lipids. It aims to test the presence of carbohydrates. It aims for test the presence of reducing sugar. You have to know this and you have to study them very well. Before moving to activity 2, you have to keep in mind that digestive juices, all of them, they contain digestive enzymes, except for the bile. What are enzymes? Enzymes are the biological scissors that digest or break down or cut or transform or hydrolyze. All of them, they lead us to the word digest. Digest food. They are biological catalysts that speed up the chemical reaction without being consumed and remains unchanged at the end of the reaction. Moving to chemical transformation of food, which is activity 2. Chapter 1, activity 3. Enzymes are the agents of digestion. So you have to keep in mind here, optimum temperature for the working of enzyme is 37 degrees Celsius. Enzymatic activity is best at this one. That's why it's called optimum temperature. Inactive temperature is 0 degrees Celsius. Since enzymes, they are inactive at this temperature, activity is 0%. 60 degrees is the denaturation or the destruction temperature. Since enzymes are destroyed under high temperature, so they can no more act. The activity is going to be 0, even though we return them to the normal temperature, which is 37 degrees Celsius. You have to study here the table of digestion in organs. Every enzyme, on which substrate it acts, at which pH, at which temperature. If an enzyme is inactive at cold temperature, but is replaced at normal temperature, which is 37 degrees Celsius in this case, in human body, so it becomes functional again. While if an enzyme is placed at very high temperature, then replaced at normal temperature, let's say very high, about 70 degrees Celsius for example, and then replaced at normal, it won't function. Why? Because high temperature will denature the enzyme. So the enzyme is denatured, it can't function again. We have to work about the mode of action of some of the enzymes. Mode of action by simplification of salivary release. Pay attention to this. You have to give legend. You have to give the legend. This is all. This chain is starch. Every one of these, every two, let's say it's maltose. Every one of these is glucose. You have to give the legend. This symbol is for the enzyme salivary amylase. This one is for the molecular simplification of proteins by the enzyme pepsin. So every one of these, every triangle is amino acid. This enzyme is pepsin. and every 3 they go for polypeptides, or every 4, every 5, and so on. So you have to give legends, you have to draw it using a pencil, you have to give it a title, and you have to write the legend in a clear way, and you have to use it. Third, molecular simplification of peptides by trypsin, the same idea. You have to give legend, you have to give the symbol for the enzyme, let's say, and it's not, should be like this, can be arrow, normal arrow, 2 arrows, can be, for example, like this. You can draw whenever you want. You can draw it simply like this. This is an arrow. You can use whatever you want. I use different shapes and different. This is going also for the simplification of lipids by the enzyme lipase, also the same idea. The simplification of maltose by maltase, this is the easiest one, the enzyme maltase, and this is glucose, so maltose is 2 glucose together connected. Moving to chapter 1, activity 4, from food to nutrients. If our digestive system is capable of digesting the food we eat, no matter what, and transform it into simple nutrients, then why do they insist we have to chew our food well before swallowing? It's easier for the digestive system to digest small pieces than digesting large pieces. This is the big answer here. So it's easier to digest small pieces than digesting large pieces. So digestion can be divided into mechanical and chemical. Mechanical involves digestion of food into smaller pieces. Chemical involves the action of enzymes. So mechanical digestion is the digestion or the breaking down of food into smaller pieces. This will keep food moving through the long digestive tube. Organs where mechanical digestion involves. Mouth, we have something called chewing of food. And the level of the stomach, we have called churning. Small intestine, we have something called peristaltic movement. Chemical digestion. Chemical digestion mainly involves the digestion of food by the help of the digestive juices. They break down food into simple nutrients. It occurs in the mouth, in the stomach, and the level of the small intestine. How are these two types of digestion needed? Mechanical digestion, you have to keep it in mind that mechanical digestion facilitates, this is the big conclusion here. Mechanical digestion facilitates, so digestion of food, or the cutting of food, or the breaking of food into smaller pieces is going to facilitate the action of the digestive juices and the enzymes present in these juices to digest food faster. The routes of nutrients. The routes of nutrients, activity 5. So we have to study the characteristics that favor intestinal absorption. We have thin wall of intestinal cell, large surface for exchange, highly vascularized, meaning that contain rich in blood vessels and lymphatic vessels, and finally its moisture. What are the different routes of absorption? We have blood, we have lymph. Blood, it absorbs all the nutrients except for fatty acid and glycerol. While the level of the lymph, it absorbs only fatty acid, or mainly fatty acid plus glycerol. Pay attention to these notes. Pay attention to the notes that I covered in this presentation. To move into the second chapter is about respiration. What is respiration? To move, we're going to talk about the respiratory system. What are the different parts of the respiratory system? What is the function of the respiratory system? And what are the different functions they do? First, we have to know that the organization of the respiratory system of humans, which is chapter activity 1. We have here the nasal cavities, the mouth, we have the pharynx, here is the pharynx, we have the larynx, trachea, lung, here we have the bronchus, the bronchioles, these are the bronchioles, the alveoli, which are called the air sacs, and we have here the muscles called diaphragm. Lung is the main organ in the respiratory system, the main organ during which respiration occurs. In this one, we have the pathway of air along the respiratory system. You have to know this pathway. We have the nasal cavities. Air enters through the nasal cavities from the nose, then to the pharynx, the larynx, then it moves to the trachea, then it moves to the bronchus, bronchioles, and finally it moves to the alveoli. What's the difference between breathing and respiration? First, you have to know that breathing is the actual intake of air. Breathing consists of two phases, inhalation and exhalation. These are called respiratory movements. Inhalation is the process of taking in air. This air is rich in oxygen, O2 gas, and it is poor in carbon dioxide. Whereas exhalation is the process of releasing air outside the body. This is poor in O2 and rich in CO2. Diffusion. Diffusion is one of the key words in this chapter. Diffusion is the movement of a substance from a medium of high concentration, of high pressure, of high volume, of high amount to a medium of low concentration, low pressure, low volume, low amount in this substance. For example, diffusion of oxygen from a medium of high concentration of oxygen to a medium of low concentration. Diffusion of a substance from a high pressure to a medium of low pressure. What is the role of the respiratory system? Respiratory system is responsible for the gaseous exchange of oxygen and carbon dioxide. These are respiratory gases with the environment. The pathway, as I said, we have the nose and the nasal cavities. Then it moves to the pharynx, the larynx. You have to study this pathway. Then it moves to the trachea, the bronchus. The plural is bronchi. As in math, you have radius, radii. Branch of the bronchus. Then bronchiole, alveolus, which is plural is alveoli, air sacs. Why are lungs very rich in blood vessels? They contain a lot of blood vessels. You have to know that lungs, they are spongy due to the many pores for the air sacs. They are light and they are elastic. Three properties: spongy, light, and elastic. We have two lungs. The right lung is made up of 3 lobes and the left is made up of only 2 lobes. Gas exchange between air and blood occurs at the level of the alveoli in the lungs, at the level of the air sacs. What are the characteristics that favor the gaseous exchange at the level of the alveoli? This question, maybe you're going to have such a question in your exam. What are the characteristics that favor the gaseous exchange? We have the large surface area of exchange of the alveoli, thin wall of the alveoli, and it's highly vascularized, meaning that it's rich in blood vessels. Which gases are the respiratory gases? This is the big question of the activity 3. Here we have the quantity of oxygen and quantity of carbon dioxide in milliliter in 100 milliliter of blood, in blood entering the lungs and in blood leaving the lungs. If you see here, the quantity of oxygen entering the lungs, it was 15, while leaving the lungs it becomes 20, so it increases. 53, the quantity of carbon dioxide in blood entering the lungs, while it's 49 in blood leaving the lungs. Here you have to compare the results of the table showing the composition of blood entering and blood leaving. Comparison, you have to talk about greater than, less than, higher, smaller, greater, smaller. We have to talk about the values here. Interpret the results of the table. You have to do analysis plus an indication. This table is showing the variation of the quantity of oxygen and carbon dioxide in milliliter for each 100 milliliter of blood and blood entering and blood leaving the lungs. The blood leaving the lungs, or blood entering, sorry, the lungs contains less oxygen, 15 milliliter, than the blood leaving. If you see here, I'm using values, so you have to use value. The blood entering the lungs contain more carbon dioxide, 53 milliliter, than the blood leaving the lungs, which is 49 milliliter. This is what's known by compare. This is how we compare a table. This is a indication. Interpret, analysis plus indication. So the quantity of oxygen in blood entering the lungs, it was 15 and increases to be quantity. To see here, I use the word increases in the analysis. So increase and decrease in the analysis, greater than, less than in the comparison. What does this indicate? This is the indication. This indicates that the oxygen diffuses. It moves a medium of high concentration to a medium of low concentration. It moves from the lungs to the blood. The quantity of carbon dioxide in blood entering the lungs was 53, and then it decreases. This is the key word here, decreases. And here we have the key word here, which is increases. So you have to be careful for these words. This indicates that carbon dioxide diffuses from blood medium of high concentration of CO2 to a medium of low concentration of CO2, which is the lungs. Here the lungs, blood, carbon dioxide and oxygen, they move from medium of high concentration to a medium of low concentration. Oxygen is diffused from the lungs to the blood, and the opposite is true. Carbon dioxide is going to move from the blood toward the lungs. In oxygen, while the blood leaving is rich in oxygen. This is a figure showing the alveoli in the lungs. Here we have oxygen gas entering and the carbon dioxide is leaving outside. Question number 2. Describe the gaseous exchange with blood at the level of the alveoli. Schematized, you have to draw a figure showing this. Oxygen diffuses from lungs. You have to study this. This is very important. Oxygen diffuses from a medium of high concentration, which is lungs, rich in oxygen, to blood, which is poor in oxygen, while carbon dioxide diffuses from blood, which is rich in carbon dioxide, to lungs, which is poor in carbon dioxide. Schematized, already have a figure that's showing schematized like this. Oxygen, diffusion of carbon dioxide toward blood, or from blood toward the alveoli. Question number 3, construct a histogram showing the variation of the composition of the blood entering and blood leaving. So you have to talk about here about the oxygen and about the carbon dioxide. Blood leaving, blood entering, you can make, for example, this is blood entering and this one is for blood leaving, and so on. Question number 4, describe the respiratory gas exchange with blood at the level of the tissues. We know that oxygen diffuses from blood, which is rich in O2, to the cells. They are poor in O2. While CO2 diffuses from cells, which are rich in CO2, to blood, which is poor in CO2. What favors, question number 5, the gas exchange at the level of the capillaries? Large surface area of the capillaries, thin wall of blood vessels, slow circulation of blood in blood capillaries. These are the 3 characteristics that favor the exchange at the level of the capillaries. Transport of respiratory gases, activity 4. You see here in this figure, we have this figure showing the transport of the respiratory gases and the different colors in this. How are these gases transported by blood? This is the biggest question here. We're going to answer this in the coming slides. But before we move into this, you have to know that blood entering lungs had dark red in color. Why? Because it is poor in oxygen. While leaving the lungs, it's going to be bright red. Why? Because it contains a high amount of oxygen. It's rich in oxygen. Alveoli, oxygen diffuses, carbon dioxide is going to move toward them. Here, this figure showing the composition of blood is composed of 55 plasma, 1 less than 1% white blood vessels and platelets, and 45% it's the red blood cells. Plasma, mostly water, they carry dissolved molecules and plasma proteins for clotting. Red blood cells, they transport oxygen and carbon dioxide. They are bound to hemoglobin. This is the idea. I'm going to talk about this in the coming 2 slides. They have no nucleus, they can't repair themselves, they have a 120-day lifespan. White blood cells, immune cells. What is hematocrit? Is the percentage by volume of red blood cells in blood. This figure showing some characteristics. They are disc shaped, enucleated, 5 million red blood cells per millimeter cube, and they contain a protein which is called hemoglobin of the symbol H capital B small. and hemoglobin can bind either to O2 or to. How are the respiratory gases transported by hemoglobin? We have different forms of the transport of oxygen gas and also different forms of transport of carbon dioxide gas. Forms of transport and oxygen and carbon dioxide. First, oxygen is transported into two forms. The first one is in the form of oxyhemoglobin, HbO8. 98% of the oxygen is transported in this way in the form of oxyhemoglobin. Also, they can be transported in the form of dissolved oxygen in plasma, which represent only 2%. So the most of the oxygen is transformed in blood in the form of the oxyhemoglobin. They are transported in the form of oxyhemoglobin. While carbon dioxide is transformed into three forms. While oxygen in two forms, carbon dioxide is in three forms. First, carbohemoglobin, HbCO2. Second, in the form of carbon dioxide dissolved in plasma, only 10%, while most of the carbon dioxide is transported in the form of bicarbonate and dissolved in plasma, 70%. And the medium that's rich in oxygen, like the lungs, hemoglobin is going to bind to oxygen. Word equation: oxygen plus hemoglobin is going to give oxyhemoglobin. Here we have 4 oxygen. You have to be careful for this. Chemical equation: 4O2 plus hemoglobin Hb, they're going to give HbO8, oxyhemoglobin. While in the medium that's poor in O2, HbO8 hemoglobin is going to dissociate into oxygen and hemoglobin in order to release oxygen and to have oxygen, since there is a deficiency in oxygen. So oxyhemoglobin is going to give 4 oxygens plus hemoglobin. The word equation, the chemical equation is HbO8 is going to give 4O2 plus Hb, the opposite of this. By this way is the transportation of the oxygen. Here the general equation: 4 oxygen plus hemoglobin. If a medium is rich in oxygen, they're going to give oxyhemoglobin. If a medium is poor in oxygen, oxyhemoglobin is going to give hemoglobin plus oxygen. So when the medium is poor in oxygen, oxyhemoglobin is going to give oxygen plus hemoglobin. This is the general equation. On a chemical equation, we have 4O2 plus hemoglobin give HbO8 in the medium of rich, while in medium poor, HbO8 gives hemoglobin plus 4O2. This is the general reaction between oxygen and hemoglobin. You have to know that the reaction between oxygen and hemoglobin is a reversible reaction. Reversible meaning this is an unstable, as hemoglobin binds oxygen in a medium rich in oxygen and releases it in a medium that's poor in oxygen. Now, we're going to move to the medium that's rich in CO2. Medium that's rich in CO2, tissues like the tissues, Hb hemoglobin binds carbon dioxide. So carbon dioxide with hemoglobin gives something called carbon hemoglobin. Carbon hemoglobin, this is the word equation. Chemical, CO2 plus Hb is going to give HbCO2. While in the medium that's poor in CO2, like the lungs, HbCO2 dissociate and releases CO2. Word equation: carbohemoglobin gives carbon dioxide plus hemoglobin. Chemical equation: HbCO2 gives CO2 plus hemoglobin. General equation: medium rich and a medium poor. Medium rich, carbon dioxide plus hemoglobin is going to give carbohemoglobin. Medium poor, carbohemoglobin is going to give hemoglobin and carbon dioxide. Chemical equation: CO2 plus Hb in the medium that's rich in CO2 is going to give HbCO2, while the medium that's poor in CO2, HbCO2 is going to dissociate to give hemoglobin plus CO2. So, as we said, the reaction is also reversible, meaning that it's unstable, where hemoglobin binds carbon dioxide in a medium that's rich in carbon dioxide and releases it in a medium that's poor in carbon dioxide. Here we have this pathway in this figure where the blood flows through the lungs. The hemoglobin and the red cells bind oxygen. Hemoglobin and oxygen bind to form something called oxyhemoglobin. Then the blood flows in very little oxygen in the form of oxyhemoglobin breaks down and releases oxygen. Then the oxygen is going to diffuse into the cells of the body. Hemoglobin in the red blood cells goes back to the lungs to collect and to pick up more oxygen to form also oxyhemoglobin and so on. So as a conclusion, this is the gas exchange in the alveoli. You have in the atmosphere, this is O2 by inhalation to the lungs, and then the lungs is going to bind oxygen with hemoglobin to give oxyhemoglobin by red blood cells or free in plasma. Then it's going to move into the tissues, CO2 free in plasma or in the form of bicarbonate HCO3- or in the form of carbohemoglobin HbCO2. Then they're going to be released into the atmosphere by exhalation. So reaction of carbon monoxide. Carbon monoxide is known to be a gas that's found in smoke. It binds in a irreversible, meaning that it's stable. It can't happen in both directions. So this binds to hemoglobin and prevent the binding of oxygen and carbon dioxide. As such, less oxygen will be delivered to the body cells so they can produce less energy, and carbon dioxide will accumulate, leading to intoxication. The reaction is HCO plus Hb, it's HbCO. This is very important note. You have to pay attention to this note. Compare the aspect of the blood entering and the blood leaving the lungs. Blood entering the lungs is dark red in color, than blood leaving them, which is bright red. Since entering the lungs, blood is poor in oxygen, poor in oxyhemoglobin, while leaving the lungs is rich in oxygen, which means that it's rich in oxyhemoglobin. That's why it has a bright red color. Compare the aspect of blood entering the organs to that leaving. Blood entering the organs is bright red in color, than blood leaving the organs, dark red in color. Since entering the organs, it's rich in O2, abundant amount of oxyhemoglobin, while leaving the organs, blood leaving the organs is poor in oxygen, which is poor in oxyhemoglobin. How many types of circulation? We have two types, systemic and the pulmonary. Pulmonary is called the small circulation. Small circulation is blood between the lungs and the heart. It aims to oxygenate blood and get rid of CO2, while systemic, it's circulation of blood between the heart and all the body organs. It aims to transport oxygen and nutrients to body cells and to drain away carbon dioxide and the waste. So the main aim of pulmonary to oxygenate blood and get rid of CO2. Okay, pay attention to these notes. And we have two types of reactions in the body cells. We have oxidation reaction and assimilation reaction. Oxidation use oxygen and nutrients, basically glucose, to produce energy. Waste, carbon dioxide, and water vapor are released during the oxidation reaction, while assimilation, they use nutrients and energy to produce or synthesize new organic like proteins and carbohydrates. Please pay attention to the notes about the worksheet, and you're gonna have a question about respiration that's already solved in your class, maybe in the exam review worksheet or in the respiration worksheet. So study these two sheets very well. You're gonna have a question which is about 5 or 6 marks about respiration that's found in this two worksheets, one of these worksheets. So pay attention to this, study well for your exam, and get ready for this exam. And it's not easy, you can do it. Thank you. Bye bye.